Hello, everyone. This is Dr. Hira, and our today's discussion is about oxidative phosphorylation. So, before talking about and explaining what the oxidative phosphorylation is, we'll talk about uh, the different types of phosphorylation and what the phosphorylation is, and what are the advantages and the significance of this phosphorylation. So, the very first thing is which may come in your mind that what is phosphorylation? So, basically, the phosphorylation is the formation of ATP from ADP, the coupling of ADP with the inorganic phosphate, and it results as a phosphorylation because you are adding up this phosphate group and one of the species, which is ADP, which is adenosine diphosphate, it gets phosphorylated by another inorganic phosphate group. So this is called the phosphorylation. And the very important thing about this, that this is a covalent modification. This is not an hydrogen bonding. This is not an electrostatic interaction. This is just not an a van der Waal um, through the van der Waal forces. So this is a proper covalent modification because here this is the covalent bond which is forming, and this is linking up this phosphate group with the ADP, and this is becoming a part of it. And now this is converted into the adenosine triphosphate. So the covalent modification is basically to modify a substrate or enzyme activity by covalently shift functionality from one molecule to other substrate. And that substrate could be the protein or an enzyme. As a chemistry student and as a minor or major chemistry student, so you know the methylation, the sulfonation, acetylation, or generally the alkylation. So these are when we are modifying the species by either alkylation or adding up the sulfonate group or sulfonyl group, and there is acetyl group addition. So this is through the covalent bond, and this is called the covalent modification. Now, this is established that in a phosphorylation, we are adding up an inorganic phosphate to the adenosine diphosphate, and eventually we are resulting in a high energy molecule, which is ATP, right? So, and which is called the cellular currency as well. So now there are three types of phosphorylation because nature has manufactured everything so beautifully that if some of the metabolites or intermediates are not there, or if there is not the surplus of oxygen, so that metabolic um, uh, pathways have to go on, that catabolic activities have to perform. So there are the different types of phosphorylation because phosphorylation is important, and we'll see in the next slides that what are the major important reasons of the phosphorylation. So phosphorylation has to perform for the functioning of the cellular activities. So there are the three types of phosphorylation. First is the substrate phosphorylation or the substrate level phosphorylation. The second one is the photophosphorylation, and the third one is oxidative phosphorylation, which is our key emphasis for this discussion. We will discuss one by one to see what are the differences and what are the key mechanism for each kind of this phosphorylation. So the very first is the substrate level phosphorylation. So for the complete one line um, concept of the substrate level phosphorylation is that this is the enzymatic transfer of phosphate from substrate to ADP to form ATP, right? So, in other words, you can illustrate that there is a formation of ATP by the transfer of phosphate, but that phosphate group is directly shifting or transferring from that substrate, and this transfer will perform by the enzymatic activities, right? So, this is basically when there is a high direct transfer of high energy phosphate group from a phosphorylated intermediate metabolic compound. So this is an exergonic catabolic pathway. And you know, exergonic uh, catabolic pathway means that there is a release of energy, right? And this is giving up to the system that energy. So here you can see this is an enzyme, and that enzyme is the pyruvate kinase enzyme, which is performing this phosphorylation activity from this substrate, which is the phosphoenol pyruvate. And you can recall that where you hear about this PEP, this is a PAP, we have discussed that in our glycolysis, right? So the phosphoenol pyruvate, its phosphate group, is directly transferring to the ADP with this enzyme activity, and this exergonic activity has performed, and there is the formation of this ATP, which is generating the energy, right? So this and then eventually we are getting the product, which is a pyruvate one. So this is the subject level phosphorylation. Now, what are the different common examples of the substrate level phosphorylation? So we can just recall that one because we already have discussed these one in citric acid cycle and glycolysis. So in glycolysis, phosphate group adds on ADP to perform ATP. In citric acid cycle, GDP plus PI forms GTP by simple conversion, and eventually it converts into ATP. Conversion of phosphoenol pyruvate to pyruvate, because there is the phosphorylation, that there is a phosphate group which is eliminating and which is just kick off from the phosphoenol pyruvate, and it is converted into the pyruvate by giving up this phosphate group. Conversion of glucose to phosphoenol pyruvate during glycosylation, where this add up that phosphate group and it converts into the phosphoenol pyruvate. So these are the common examples of substrate level phosphorylation. Now the next is photophosphorylation. So as the name state, this is linked with the light assisted mechanism. So the formation of ATP in a light reaction, and the most best example is the photosynthesis. So in which the photosynthetic organism capture energy of sunlight, the ultimate source of energy in the biosphere, and then harness it to make ATP. Photophosphorylation involves the oxidation of water to oxygen with NADPH positive as ultimate electron acceptor. It is dependent on the energy of light. So if you minus that light source, so this phosphorylation is not going to happen. Or either you're not providing that source, which is the electron donor and acceptor. So even then, in that case, that's not going to happen. But the initiating step, as we do recall that what is initiator in some of the reaction activities. So this is the sunlight, this is the energy which is providing it's the motivation or it's rational for conducting this photophosphorylation. Now in this slide, this is to give you the idea that what is happening in a photophosphorylation. So just look at this one. We know that in a plant cell there is a chloroplast, and in chloroplast we know these um, substances, which is the chlorophyll, right? And this chlorophyll has the capacity to absorb that light energy. It take up this water molecule from the soil, and then there are the removal of the electron. So this is basically the water breaking of that water molecule into 2H positive and into the nascent oxygen. So this can break up into this one, and there is elimination of the two electrons, and eventually these electrons are becoming the part of this electron transport chain. Well, there are number of steps, and there are a number of positions of the functionality where that electron capture, then this oxidize, reduce, deoxidize. So that is happening. So there is the chain of that steps. So we will discuss an electron transport chain later. But right now, I'm just giving you the idea that this is not a single step. So there are multiple steps by which these electrons are transferring from one step to another, second, third, fourth, and eventually this is coming up with NADP positive and converting into NADPH. And there is a removal of the ATP, which is, or basically the target moiety, or the target, or you can say the overall product of the phosphorylation. This is the image to give you that N or biosystem, how this is happening, how this light is becoming the part of this photosystem. They are specialized electron chain carrier proteins, right? And then they are splitting that water molecule into the H positive, and this is creating some kind of gradient where that is move on, right? And but this all happening in the presence of that light. So this is the photophosphorylation. Now the next is, what is oxidative phosphorylation? So the oxidative phosphorylation is a metabolic pathway by which energy released by the degradation of food components and is stored in adenosine triphosphate. ATP is produced in all living organisms having aerobic oxidation. So this is the important point, that we are talking about the aerobic oxidation, where there is a presence of oxygen is necessary. So this is not the oxygen minus environment. So this is where an environment where there is the um, presence of surplus presence of oxygen. So although they may have different sources of energy, NADH or FADH2 dependent ATP synthesis is the result of oxidative phosphorylation. So, this is an example of the oxidative phosphorylation. Formation of ATP from the oxidation of NADH or FADH2 in presence of oxygen through electron transport chain is known as oxidative phosphorylation. So, you can mention that definition in that way. All oxidative steps in the degradation of carbohydrates, fats, and amino go through this final stage of cellular respiration, in which the energy of oxidation drives the synthesis of ATP. And that is our target, where we started from discussing the cellular respiration. Then we talk about the anabolism, catabolic steps. And we started with the glycolysis. We talk about the Krebs cycle, and we come up that there are the number of other fuel sources along with the carbohydrates. There is the proteins, there are the fats, which do contribute to the acetyl CoA, and then they are becoming the part of the Krebs cycle. So from the glycolysis, we end up at the, we were talking about that the carbohydrate convert into carbon dioxide, and then there is eventually the other metabolite formation. And the final target is that there is the need, the body, the biological system, the different metabolic cycles, so they require the energy to move on, to carry on its movement, right? So this ATP is uh, producing in that amount by which we are performing or activities. This is not the byproduct amount of that ATP, which we have seen in number of steps, but this is basically producing, and this is the ultimate step in in the cellular respiration, where we are getting finally the ATP, right? So the oxidative phosphorylation is a redox reaction in which electrons are transferred from donor to acceptors. This redox reaction releases energy, which is used to produce ATP, because ATP synthesis is basically the an endergonic reaction. Now we will see electron stored in the form of the reduced coenzyme NADH or FADH2 are passed through an elaborated and highly organized chain of proteins and coenzyme therefore called electron transport chain carriers, or simply the electron transport chain, or the carrier proteins. So finally reaching oxygen, molecular oxygen, is a terminal electron acceptor. So do remember that finally what is our ultimate electron acceptor is basically the oxygen, which has to eliminate um, uh, and there is a production of ATP, right? So, as I have shown you in the previous slide in the photophosphorylation, that there are a number of steps in an electron transport chain. And similarly, this is happening in the case of oxidative phosphorylation, that this is not a single step. There are a number of metabolites which are taking up these electron, and there is oxidation, reduction, and reoxidation. So, by this transport chain, this is happening. So, each component of the chain can exist in at least two oxidation states, and each component is successively reduced and reoxidized as electrons move through the chain from NADH or FADH2 to oxygen. So, obviously, once they are becoming the NADH, then NAD positive, FADH, then FADH2. So eventually they reach their destination, the oxygen one. In the course of electron transport, a proton gradient is established across the inner mitochondrial membrane. That we need to know that this oxidative phosphorylation is basically happening in mitochondrial membrane. So there are the mitochondrial matrix, there is the mitochondrial inner membrane, there is the interspaces. So we'll talk about this, and this is basically happening in the inner mitochondrial membrane. It is energy of this proton gradient that drives ATP synthesis. So this energy, the initial energy which is required for the synthesis of ATP, because this is um, uh, anagonic step or activity. So this is coming when the proton is transferring from one membrane to another one, and there is a gradient. The concentration difference has established. So that is giving the energy which drives ATP synthesis. So in eukaryotes, oxidative phosphorylation occur in inner mitochondrial membrane, and in prokaryotes occur in plasma membrane. That makes sense because in eukaryotes we have the specialized cells, organelles, and they all are well wrapped with the specialized membranes. There is a proper mitochondrial structure, but in prokaryotes these are simpler cells, and there is a plasma and there is a nucleus only, and then there's this is happening in a plasma membrane. Oxidative phosphorylation involves the reduction of oxygen to water with electron donated by NADH and FADH2. It occurs equally well in light or darkness. So it means this is not dependent on the light. So if you're supplying light, it is happening. If it is dark, even then this is happening. So this, because this is independent of the light source, because this is not the photo assisted mechanism which is happening. So, why phosphorylation is important? So, covalent modification through phosphorylation is most efficient, common, convenient, and effective because of the following reasons. Um, because of this, there is a net negative charge when it is attached with any of the protein or the enzyme residue. This gives the excessive negative charge and thermodynamically stable compound, right? From ATP, which is high energy and comparatively unstable than the ADP. So when it forms in an ADP, so there is a phosphate group which get attached to some of the residue, and this is giving the negative charge, which make it more feasible, more efficient for establishing the hydrogen bonding with the number of substrate active sites and increase the specificity interaction of enzyme with substrate molecule. So this is a mechanism which the nature has developed, that this phosphorylation, when it is happening in a biological systems, so this is providing the interaction or the bindings of the enzyme with the substrate. ATP Abundance, ATP is present abundantly within cell, which can easily use for phosphorylation. And the phosphorylation is reversible, so this is not irreversible reaction that if once it happens with the ATP, so we can't go back. So this is a reversible reaction. There's ADP formation, then eventually as per requirement, this converts into the ATP one. So next is oxidative phosphorylation cycle. So this is what exactly happening and what kind of production or the yield we are having in oxidative phosphorylation cycle. So basically, we can break up this oxidative phosphorylation into as a couple of reaction or the two main um, uh, chapters under two main umbrellas or the reaction types. So the first is the exergonic process and the other one is the endergonic process. So exergonic process is the reaction electron transfer from electron donor to acceptor take place, which is exergonic process, right? So if there's electron which is accepted by the acceptor one, so there is a certain amount of electron energy which required to eliminate that electron from that species and then uh, uh, towards the that acceptor side. The energy released in the above process is used to synthesize ATP from ADP, which is endergonic process. NADH is common electron donor and oxygen is common electron acceptor. So that you have to keep in your mind that in oxidative phosphorylation, so what is basically the ultimate electron donor, and that or the key electron donor is NADH, and the common electron acceptor is on oxygen. This electron transfer and ATP synthesis are taking place in a membrane, and the energy transfer takes place by transfer of protons across the membrane. We have seen, discussed in a previous slide, that there is a gradient or the concentration gradient which is established by the movement of the proton from one membrane to another, inner, outer to inner, or inner to outer. So this gradient is establishing the energy difference, and that is basically facilitating for the synthesis of ATP. This transfer of proton across the membrane due to energy difference is called a chemiosmosis. The effective yield of oxidative phosphorylation is much more higher than an aerobic respiration process, right? Because as we know, this is oxidative phosphorylation, and the oxygen is a common electron acceptor. So when we have the surplus supply of the oxygen, so this chain is continue, right? And there's more number of ATPs we get in oxidative phosphorylation rather than in the system where there's absence of oxygen, and we termed it as an anaerobic respiration process. So there is a limited moles of the ATP which will be produced because oxygen is not available as a common electron acceptor. So for example, glycolysis produce only 2 ATP, which is not the oxygen dependent mechanism, while oxidative phosphorylation produces around 30 to 32 ATP molecule, right? Here you can have a comparison, and this is a drastic difference that 2 versus 32 or 30. So here's a huge number of ATP we get in oxidative phosphorylation in aerobic respiration. Product of oxidative phosphorylation. So the complete breaking of food into energy can be studied under 2 steps. So energy wise, we have divided it exergonic, endergonic, but what actually is happening is that the first thing is there is a breaking of glucose to give pyruvate and carbon dioxide cycle. Then the second is the synthesis of ATP with the help of reducing agents already produced. So in the first step, there are further sub steps which I will discuss with you. So in a breaking of glucose to give pyruvate and carbon dioxide, the what is happening very firstly, all the energy producing components of our food are finally broken into glucose and easily digestible form. Glucose undergo decomposition to give pyruvate ion and NADH, right? So here, this is the first, um, from the first step, this is the very first reaction, reaction one, where is the glucose, there is the phosphate, ADP, NAD positive, then we get two pyruvate molecules, to ATP, NADH, to H positive, and H2. So this 3 minus and this 4 minus is representing that how many overall negative charge we have in the single ATP and in single ADP. So in a single ADP, we have the -3, while in ATP, this is the -4. In the above step 2, ADP molecules are converted to ATP molecules. Now, the next step is the pyruvate formed, which we have formed here, is converted to acetyl CoA. And in this step, two NADH are produced. And now you can recall that these both step was the part of um, or glycolysis. When we talking about the glycolysis, so the both belongs to the glycolysis cycle. So the pyruvate plus acetyl coenzyme A plus NAD positive, then there is acetyl CoA, and there is the NADH plus carbon dioxide. So we get up with the carbon dioxide because in the glycolysis, the product, the major, the ultimate product was the carbon dioxide. Now the next is in the third step of the one major step, which is a breaking of the food into the carbon dioxide. This acetyl CoA, then plus 3 NAD, FAD, GDP, phosphate group, water, and it eventually convert into the carbon dioxide. The more molecules of the carbon dioxide we got there, and NADH, FADH2, which are their reduced forms. Then GTP, 2H positive, coenzyme A. This cycle is called as citric acid cycle, right? And we have seen that this was from acetyl CoA as the fuel molecule or the starting point. Acetyl CoA then add up into the citric acid cycle. So this is the step which is in the citric acid cycle we have discussed. The fourth one is in the fourth important step, the reducing agent like NADH and FADH2 undergoes oxidation with oxygen, which releases energy. The energy release is used to synthesize the ATP from ADP. The overall reaction can be represented as ADP, inorganic phosphate, biphosphate plus NADH. Then there is an oxygen by the splitting of this water molecule. If you just combine this, so this is a water molecule, but this is in its ionized and split form. So this is a two proton, and there is a missing oxygen because it's just cancel it. So ATP plus NAD positive plus 2H2O. In this step, a lot of ATP is synthesized and it is responsible for high efficiency of oxidative phosphorylation over glycolysis step. So the note is the free energy change for the overall reaction is negative and hence the reaction is highly spontaneous that we have discussed in energetics of biomolecular or cellular respiration. Now, from the figure that how we can see and compare that what was the energy gradient, what concentration gradient of the proton, and how this is facilitating the production of ATP. So H positive transport result in an electrochemical gradient. So when this H positive is higher on one side and then it is moving to the lower side, so there is the gradient which is established, and this is called the electrochemical gradient. Positive, where there is abundance of the proton, and which is deficient in a proton, obviously, when they have the counter charges there. So this is a negatively charged. Proton motive force, energy released by flow of H positive down its gradient is used for ATP synthesis, and that is called proton modification. ATP synthase is an enzyme, H positive channel, that couples energy from H positive flow with ATP synthesis, and this is the basically enzyme which is facilitating this ADP to convert into the ATP1. Significance of H positive transfer is that a difference in H positive concentration is generated, which is a concentration and electrical gradient. This requires an intact inner mitochondrial membrane to maintain the gradient. An inner mitochondrial membrane impermeable to H positive. Electron transport provide energy to move H positive against its electrochemical gradient, right? Because otherwise this H positive is not able to cross that membrane. So there is the electron transport is basically mechanism, which is the chemiosmosis, which is basically shuffling or forcing this hydrogen ion to move to other side, right? So the translocation of H positive during electron transfer from here to here, and then forming the water molecule. So here you can see that OH are moving from there and this, and again these are the phosphate transporters, which is called because they facilitate the phosphate group to transport into the inner cross the inner mitochondrial matrix and then becoming the part of mitochondrial matrix. Now we'll see what are the similarities between photo and oxidative phosphorylation. So these couple of slides are just a comparison between the all three kinds of phosphorylations. So oxidative phosphorylation and photophosphorylation are mechanistically similar in three respects. Both processes involve the flow of electron through a chain of membrane bound carrier proteins. We have seen there are ladder like steps, that there are specialized proteins which are taking up these electron transfer from one step to another um, substrate, and then eventually it is becoming the part of that oxygen. So the free energy made available by this downhill exergonic electron flow is coupled to the uphill transport of proton across a proton impermeable membrane, conserving the free energy of fuel oxidation as a transmembrane electrochemical potential. And this is happening in a both way in both phosphorylation types. The transmembrane flow of proton down their concentration gradient through a specific protein channels provide the free energy for synthesis of ATP, catalyzed by a membrane protein complex, ATP synthase. This is not a single protein. There are more than 5 protein complex which are present there, and they are facilitating that one. That couple the proton flow to phosphorylation of ADP. Now, the substrate level phosphorylation versus oxidative phosphorylation. So, the breakdown of glucose molecule by glycolysis followed by citric acid cycle generates very few ATP molecules and is known as substrate level phosphorylation. And we have seen that there is only 2 ATP molecules generated when this is a substrate level phosphorylation, while in oxidative phosphorylation, this is almost 30 to 32 ATP molecules, which get synthesized in the oxidative phosphorylation. Um, in these cycles, many reducing agents like NADH and FADH2 are produced, and the reducing power can be used to produce more ATP. And that's why there is the more number of ATP production as compared to the substrate level phosphorylation. Both substrate level phosphorylation and oxidative phosphorylation involves formation of ATP. That is the similarity point. But in substrate phosphorylation, there is a direct transfer of phosphate group to adenosine diphosphate by an enzyme activity, while there is no direct transfer of the phosphate group in oxidative phosphorylation. We have seen there is the electron transfer chain. Step by step, this gate transfer from one to another, and then third and the fourth point. The substrate level phosphorylation takes place in cytoplasm, while the oxidative phosphorylation happens in the mitochondrial matrix. The major difference here is substrate level phosphorylation, the oxidation and phosphorylation are not coupled, while they are coupled, or you can just, these are the simultaneous activities which are happening, that there is a phosphorylation which is facilitated by the oxidation and the reduction process or the redox activity. So that's why this is called oxidative phosphorylation. So that was all about the phosphorylation, and I hope you got some of the understanding of the phosphorylation, the differences and the similarities, and the importance, and what is the mechanism, what are the key products. So just let me know if you have any queries and you are just be curious about some of the terms which are not making sense for you. So you can just ask the question, you can comment in a video, and I would be happy to answer you. Take care. Have a good day.